Drugs have been a enduring feature of human existence. Are you getting, is the recording okay? Can you hear me all right? Sound okay, is it? Good, good. And if you go around from the top left, you see there's peyote, used by Native Americans. One of the many magic mushrooms that exist in the world. On the right hand side at the top, there's ayahuasca being brewed up, these two plants which together allow you to drink DMT. Then Amanita muscaris, morning glory. But the most important one, I think, is the bottom left hand corner. That's a Greek vase. It's over 3,000 years old. And it shows Greek noble person partaking of a cocktail of alcohol and ergot. And the Greeks knew that when ergot started to grow on the rye, if they ate ergot and drank alcohol, they would have a very interesting experience. They called it the Eleusinian Mysteries, and they would actually spend their holidays in September, October, when ergot was growing, celebrating the mind-altering effects of those two drugs. And then after that, they'd retreat back to their city-states and do the things that they were good at, like, you know, invent geometry and music and things. So it was a very interesting example of how psychedelics may have seriously influenced the whole development of Western thought and culture. And there's also an interesting angle, and I hope I don't offend anyone who is intensely religious, but there's a credible argument that Amanita muscaris, this red-capped mushroom, may have had a major role in the development of Christianity. It seems to have been used by the early Christians to produce bonding and to enable them to confront the vicissitudes of being an oppressed minority. And here you see some examples of the mushroom in early Christian art. The one in the middle is a very famous fresco, and it basically recapitulates the tree of life, not as an apple tree, which of course didn't exist in the beginning because they were cultured subsequently, but as a large mushroom. And the left-hand side there, the very bottom left, is one of the most important books in the history of Christianity. It's the Cambridge Psalter, and the Canterbury Psalter. And it shows that on the fourth day God created plants, and most of those plants were actually mushrooms. And there was a very successful attempt in about the 12th century. The church, there was schism developed in the church. The church became a money-making establishment, and it wanted to eliminate records of this previous heathen behavior. So most of the records relating to the use of Amanita were destroyed, but not all of them. You can see there's some of the ones at the left. And that book by John Allegro is a remarkable analysis of the potential contribution that this particular psychedelic has made to Christianity. But I want to talk to you today about neuroscience. I am a sort of neuroscientist. I'm a psychiatrist, psychopharmacologist. I'm very interested in what drugs do in the brain because I'm interested in why people have mental illnesses, but I'm also therefore interested in consciousness. And this is one of the most profound challenges that have been laid down to our discipline. There's William James, the father of American psychology, one of the founding fathers of the discipline of psychology. And he used a variety of drugs, including mescaline and nitrous oxide, to explore the nature of consciousness. And he wrote this: "Our normal waking consciousness is but one special type of consciousness, whilst all about it, parted from it by the filmiest of screens, there lie potential forms of consciousness entirely different." And this is the key point for you as scientists and the future of this research area. No account of the universe in its totality can be final that leaves these disregarded. Now that is a wonderful challenge, isn't it? A wonderful challenge to science. To understand the universe, you've got to understand altered consciousness. Of course, he threw that down in about 1896. In those days, there were very limited tools for exploring the brain. And as he pointed out, how to regard them is the question, for they're so discontinuous with ordinary consciousness. But things have moved on now, and we're all the inheritors of his challenge, and I think we're beginning to answer some of the questions he raised. Of course, the great transformation in terms of bringing altered consciousness to the public domain were the writings of this man, Aldous Huxley. And he came from the leading academic family, the greatest family of scientists there's ever been, probably in the world, but certainly in Britain. And his stepbrother got the Nobel Prize in physiology and medicine. He wanted to be a doctor, and he couldn't be a doctor because he had terrible visual problems. So he went to literature and art, and his life was transformed when he took mescaline. And he wrote about it in his book, The Doors of Perception. And most people don't realize that The Doors of Perception is a quote from an earlier British writer and artist called William Blake. Blake, like many great artists, could see the world, or saw the world very differently to the way we see it. And Blake said this: if the doors of perception were cleansed, everything would appear to man as it is, infinite. For man has closed himself up till he sees all things through narrow chinks of his cavern. And Blake was saying basically that most people don't really see what the world is. They see a very narrow, limited amount of the world. And Huxley realized that what mescaline had done, and what LSD subsequently did for him, was to open up the doors of perception. And then he came to this conclusion, therefore. What's closing the doors of perception must be the brain, because these drugs work on the brain. And he said this: the brain is an instrument for focusing the mind. And that's a wonderful observation. So there's a subjective experience turned into an interpretation of what the brain does 60 years ago. And the wonder is that we now know he was right, because modern neuroscience proves him right. So for those of you who aren't neuroscientists, I'll just explain to you how the brain works. We'll use vision as an example that's perhaps the best studied. So your brain is not a camera. The way you see me is because photons come from me, they go into your eye, and the back of your eye is your retina, and your retina decodes them. It decodes all the different elements of what you see, the colors, the shapes, the position, and it turns them into electrical impulses. And those impulses travel up the optic nerve, and then they disperse through different parts of the brain. About a quarter of your brain is involved in actually organizing what you see now. And the brain puts together what it thinks is out there based on those electrical impulses. So your brain reconstructs what it thinks is out there. And the reason it reconstructs it is because it's way too energy inefficient to be continually scanning the environment and getting a detailed reconstruction of what's out there. Your brain is updating its information several times a second. To have detailed analysis of every piece of information would be just ridiculously wasteful. So essentially the brain works out what's likely to be there and says that's good enough, and that's all you see. And it's quite likely that if I was halfway through my talk to turn into a tiger, you probably would take a long time to notice, because you're still thinking. In fact, for some of you who are doubting it, I am a tiger, but so we'll come back to that another time. Anyway, the brain reconstructs what it expects to see, and it does it in this very limited way, as Blake pointed out. The brain essentially, as you're looking at me now or looking at the screen, you're not looking at anything else around here. Why bother? Because you're not interested in it. The brain really focuses down on what's really important. But there's another interesting take on this, which is that what people see is often strongly determined by how they feel. And when depressed people look out through the chinks of their cavern, they don't see the bright sky and the pretty clouds that you and I see. They see a grey, miserable environment. And when an addict looks out through their chinks of their cavern, they see their love objects, the bottle or the syringe. So what you see is strongly affected by what your brain has decided is important. And that is the reducing valve concept. So psychedelics were plant products until Albert Hofmann in the 1940s modified ergot to make a much more longer lasting version, which we now call LSD. And there's an image of him and his 100th birthday sitting in his house overlooking Lake Geneva. And I don't think his house is actually that colour, but the point is he managed to survive to 100 despite taking LSD regularly, which rather belies the concept that it fries your brain. The first psychiatrist, we think, to take LSD was Joel Elkes, and he lived to 103. So I'm not suggesting that psychedelics make you live a long time, but they don't seem to cause premature death anyway. And LSD became the revolution because of its stability, effectively, and the fact that it was very interesting. The effects in Hofmann led him to persuade Sandoz to make it available. And some very interesting people used LSD when it was legal. And these are the two I want to talk a bit about, because these are the two most important Nobel Prizes in medicine ever. Francis Crick with the Jim Watson decoded DNA. And Kary Mullis, American biochemist, was trying to find out whether there were differences in DNA which could explain why animals and plants were different. But he was struggling to get enough DNA. It took Crick and Watson a very long time to collect enough to do their crystallography. And he was trying to work out how he could get enough DNA to do experiments. And he says one day, whilst taking LSD, he saw the molecule, the double helix molecule, unraveling in front of him like a series of serpents. And he realized that that's what he had to do. He had to find a way of replicating DNA. And he hunted around and he found a bacterial enzyme called polymerase, which did that. He could make it replicate DNA. He developed the polymerase chain reaction and got the Nobel Prize, because the PCR underpins almost all life science, but also underpins most food science. The way you know, when you go out tonight, you'll be able to determine whether the burger you eat is a beef burger or a cow burger or a horse burger by PCR. PCR tells us everything you need to know about the origins of food. So it was a remarkable discovery. And Mullis says that he wouldn't have had that insight if he hadn't taken LSD. Crick's story is rather different. Crick had got the Nobel Prize for understanding the origins of life. He then helped Sanger get a Nobel Prize for working out how DNA transformed itself and decoded into RNA and proteins. And then he thought, well, what next? I've sorted out life. Is there anything else? And he started taking LSD. And his parties in Cambridge became quite famous because they were fun and also full of very influential and powerful people. But unfortunately, the drug was made illegal. And soon after, the police came to his house. Actually, it was quite easy to find because he stuck a golden helix outside. But and they said, you've got to stop having these parties because the drugs you're taking are illegal now. And like a true scientist, he said, well, that's ridiculous. And then he left. He left Britain. He came to America, where the drug was illegal, but the police didn't pester him because he had a Nobel Prize. And or maybe because he lived in La Jolla. I don't know. Maybe it was the West Coast. But anyway. And he spent the rest of his life trying to solve the real problem, which was not how life is created or manufactured or replicates itself, but the nature of the brain and mind. And I love this quote, which is attributed to Einstein: no problem can be solved from the same level of consciousness that created it. And I believe if Crick was alive today, he would agree with that, because I have no doubt that it was his use of psychedelics that made him understand the challenge of the brain. And he says this: there is no scientific study more vital to man than the study of his own brain. Our entire view of the universe depends on it. And he wrote this book, and it's a remarkable book because to us it's trivial. Yeah, that book basically says the mind's something that the brain makes. Almost no one disputes that today. But at the time, it was an astonishing hypothesis because people were still dualists. They still thought the mind was out there, the brain was in there. And he worked with a guy called Christopher Koch, and they spent a long time trying to determine the anatomy of consciousness. And they came up with quite an interesting theory. They came up with the theory that the claustrum, which is an interesting collection of cells which underpin significant parts of the cortex and seem to gate cortical access of thalamic information to the cortex. They came up with a conclusion that the claustrum was the seat of consciousness. A very difficult experiment to test because it's a diffuse layer of cells. You can't ablate the claustrum to see whether it alters consciousness. Anyway, you wouldn't ethically be able to do that in humans. In fact, they were sort of half right. Actually, the claustrum doesn't regulate consciousness generally in the way we think about it, but it does. It's almost certainly, it's almost certainly the site of the altered consciousness produced by salvia divinorum. Because salvia is a kappa agonist, and the claustrum is full of kappa receptors. So those altered states of consciousness produced by salvia, I believe, are probably due to disruption of claustral functioning. No one's done that study yet. It would be really interesting to do. Very hard to image the claustrum, but at some point, we would hope to do it and prove him right. But those of you who know about salvia, know it's not a psychedelic in the same sense as the serotonergic psychedelics we're talking about, like LSD and psilocybin. Now, LSD was such a powerful drug, and in those days the pharmaceutical industry was much more open to research than it is today, that Hoffman persuaded Sandow to make it available. There it was a medicine, see? Delicid, it was ampules of LSD. And they gave it out to about 1,000 registered scientists around the world. And it was used for 4 reasons. It was used to model psychosis. We're still doing the same today. We've set up trials recently to use psilocybin to produce states of altered consciousness, which we are trying to test new anti-schizophrenia drugs on with some success. And if you want, I can talk about that in the discussion. It was widely used by health professionals to understand the nature of altered consciousness, to help them make sense of what their patients were experiencing. And it was quite common for junior psychiatrists. On the day they started their job, they were told the next day they were going to be given LSD, and they were. And then they had these interesting experiences, and I'm trying to collect them. They're dying off now. We're talking about people in the 50s and 60s. But some of these descriptions were quite remarkable. They remember them to the day. In fact, if you've got an hour, they'll tell you every single thing that happened on that day. Remarkable how they're etched in their memories. And then it was used for psychotherapy. 2 forms of psychotherapy. Psychedelic psychotherapy, which is the sort of stuff we're doing today, quite high dose. People have a powerful trip. They strongly alter their sense of consciousness. They have a mystical or peak experience. And that was quite popular in the USA. And then psycholytic psychotherapy, which is sort of towards microdosing, but doses enough to produce an altered sensation that was given regularly to help people break down their resistances, their barriers, that analysts would say, and therefore engage better in the therapeutic process. And as you all know, and I'll talk about it in a minute, these drugs were banned in the late 1960s, but some people carried on, and here's this man, Stan Grof, who worked underground with psychedelics for a very long time. And he said this: psychedelics used responsibly and with proper caution would be for psychiatry what the microscope is for biology and medicine, or the telescope for astronomy. And hard to dispute that. So why don't we use them today? And the reason, of course, is because you were in the Vietnam War. I'm going to lay the blame at your feet. Because the rebellion against the Vietnam War, when 18-year-olds, there's none of you, you're all over 18, all the men here are over 18, you could have been sent off to Vietnam to fight in a war. Just randomly depending on what day of the month you were born. And a lot of young men were resisting this because they didn't know where Vietnam was, and they didn't know what the war was about, but they sure knew it was pretty unpleasant. And here are a couple of quotes from men fighting in Vietnam, which rather unsettled the establishment. So here's Ron Kovic, you probably know about him with his broken back and the film about him. All I could feel was the worthlessness of right here in this place and in this moment, all for nothing. And Michael O'Brien, I think, said, I did not want to die, not ever, but certainly not then, not there, not in a wrong war. And the anti-war movement started, and LSD was seen as a major fuel of that movement. And it's just a few images of that time. People who were escaping the draft would sometimes go to San Francisco and drop acid, listen to the Grateful Dead and put flowers in their hair and then protest the war. Timothy Leary became the most feared man in America because he was encouraging people to take psychedelics and to drop out of society. But the most important one is the right-hand one. This protest against the war, saying drop acid, not bombs. I actually kind of feel that might well be a sentiment for our time. I think we wouldn't have 6 million Syrians destroying the balance of politics in Europe if we hadn't allowed the Syrian war to turn into the chaos it was. But that was too threatening. The idea that you couldn't bomb your way to success was something that was anathema to the government at the time, and they thought LSD was a problem and they had to get rid of it. But in those days, you couldn't just ban a drug because you didn't like it. You had to create evidence of harm. And since there was no evidence of harm, the government and the CIA had to create evidence of harm, and they colluded with a number of media outlets to create these stories. Now, it's hardly, I don't want to give them the term of newspapers, because as you know, they're not, but these completely fictitious and absurd headlines were enough to scare the populace. The populace are, the people that read these aren't likely to be very interested in the underpinning science. They would only read these to fulfill their own prejudices usually. And this was seen as enough. The newspapers are scared about LSD. There's public concern. We have to ban this drug. And what's really remarkable and chilling about this ban is that the ban occurred in the face of opposition from the most powerful man in America, the Secretary of State, Bobby Kennedy, the man who would have been president if he wasn't assassinated. And his bureaucrats are telling him that the last 15 years of funding research on LSD was all a mistake. There is no value in having LSD as a research tool. And he's saying to them, look, if these LSD projects were worthwhile 6 months ago, why aren't they worthwhile now? We keep on going around and around. If I could get a flat answer about that, I'd be happy. Is there a misunderstanding about my question? He knew he was being lied to. I think perhaps we've lost sight of the fact that LSD can be very, very helpful in our society if used properly. And yet even he could not stop these drugs being banned, because the bureaucratic process of banning drugs is inexorable. And I will argue that that ban is the worst example of the censorship of research in the history of the world. I can only think of one possible comparable example. You've got to go back a long time. You've got to go back to 1616 when the Catholic Church banned the telescope. And that wasn't a worldwide ban because Northern Europeans, the Protestants, were allowed to study the skies. That ban lasted 150 years, 140 years. The ban on psychedelic research has lasted 50 years and is still going. The amount of research that could have been done in the last 50 years is vastly greater than the lost research from the banning of the telescope. because neuroscience and psychiatry is a much, much bigger discipline. So I don't think there's any doubt that this ban is the worst ban in the history of science. When you talk to the regulators, they say, no, we didn't ban research on these drugs. We just put them into a very safe place in the conventions and the drug laws, just to make sure people didn't misuse them. But of course, de facto, it was a ban, because until 1967, there were thousands of papers published. And since 1967, the number of papers on this topic have massively declined. And here's an analysis that was published just a year or so ago. So this is the number of papers each year, either on LSD in blue or psilocybin in red. And the arrow shows when the ban came in. So you can see the massive growth, up to 250 papers a year on LSD till it's banned. And then there's a few years when people carry on writing up the results of their research, and then it collapses down to virtually none. And the reason for that, of course, is that it's impossible to get the drug to work with. There's never been a study in America since 1967 on LSD. Before then, there were 1,000 papers published from America, not one since. Now, whether you say that's not a ban or not censorship is, of course, irrelevant, because the reality is there is no research, and it's outrageous. The opportunities have been lost. If you're interested in the history and the politics behind it, I've written a couple of papers. The top one's a very big, detailed review that covers not just psychedelics, but also cannabis and MDMA. And the bottom one's a slightly simpler one. That's also a freebie if you want to get that from PLOS Biology. Well, about 15 years ago, we decided to start fighting back, and it's just over 10 years. 2 weeks ago, I gave the first injection of psilocybin to someone in a brain scanner, and that was 10 years ago. So we've been fighting back with the help of a charity called the Beckley Foundation, and now other charities. And it seemed to me pretty much all my life as a psychopharmacologist that probably the most interesting experience that the brain could undergo was the psychedelic experience. But it had never been studied. And so we decided it was time to study it. We started off using psilocybin, the active ingredient of magic mushrooms. There's some Liberty caps there. At the time, psilocybin was so expensive to buy, we couldn't give it orally. We had to give it intravenously, because that way you reduce the dose tenfold. We just didn't have any money enough money to do it orally. And psilocybin, like the other psychedelics, are all on this axis here, which is an axis of 5-HT2A receptor affinity. At the very top here you have LSD, which is very high affinity. At the bottom here you have mescaline, which is low affinity. And here you have psilocybin, which is moderate affinity. So it works on the receptors we expect it to work on. It's relatively short acting if you give it intravenously. And another major reason for using it is that no politician or newspaper journalist knows how to spell or say psilocybin. So they get a bit twitched about trying to take you on in public. Why was I interested in these receptors? Well, the 5-HT2A receptor is a fascinating receptor. There are more of these receptors in your brain than the brains of any other animal. They're in the most highly evolved parts of the brain. This is a heat map of the receptors. You can see they're in the areas of the brain, posterior cingulate, anterior cingulate, the high-level functioning areas of the brain. They're not in motor cortex, and they're not in the deep brain structures like thalamus. These receptors have evolved with human brain. Some people believe they have caused the evolution of the human brain. This great mystery as to why our brains are so much bigger than other primates. Some people think that that's been driven by these receptors, and it's not implausible that is true. And 30 years ago, I did the first study with a pure antagonist of these drugs. I gave a drug called ritanserin. I blocked all these receptors in humans, and nothing happened. Except that when we recorded their sleep, they went into a deep sleep, which had, with a great deal of synchronicity, in terms of the slow waves of sleep. So it's a kind of paradox. You block all these receptors, and yet nothing really happens. You can still go to work. I took it for 3 weeks. It was fine. So why are they there? Well, of course, the only way you can really answer that question is to stimulate them. And that's what psychedelics do. So we got round to this experiment of stimulating them. But I want to say a little bit more about the brain first, because how many of you are neuroscientists? Okay, and so the rest of you, listen up. So the way the brain is a very sophisticated collection of neurons. Your brain has 200 billion neurons, of which 160 billion are these pyramidal cells. These neurons, like here, that have their bodies here and stick their dendrites up through here. So most of your brain is just a mass of pyramidal cells. Your cortex has a whole series of different layers of pyramidal cells, but this particular layer here is where most of the 5-HT2A receptors are. And the way the brain works is that each of those pyramidal cells is like a computer. It does the primary analysis of whatever is going on in the brain. And that's done vertically, up these things called cortical columns. And then the secondary analysis, the secondary analysis, bringing together all the different individual analyses in the brain, is done by these cells, which communicate across the brain. So the 5-HT2A receptors are on the neurons which integrate brain function. And we presume that's why they're there. And these neurons are so important that they have a special set of what are called interneurons, GABA interneurons, to regulate their activity, so-called chandelier cells. Okay, so there you have it. The brain is this two-dimensional computer. And what happens then when you stimulate these receptors on the neurons which communicate across the brain? Well, we did the experiment, and then we analyzed. So the first experiment we did was using a technique in fMRI called arterial spin labeling. It looks at blood flow. What has happened here? And to our amazement, we discovered that despite the fact that those subjects were having powerful visual hallucinations, they were having powerful alterations in the sense of self and body. Some of them described their body atomizing and going outside of the scanner. One went to heaven and met God. He came back, which was good news. These powerful experiences, we expected to see increases in brain activity, but when you see these images, blue means less. And we were staggered. How could you have all these experiences with the brain, bits of the brain being shut off? And it was these three parts of the brain that are being shut off. And it's interesting, you'll notice already that the two parts of the cortex that are shut off are the parts with the high density of these receptors. And in fact, there was a good relationship. The more you shut off the anterior cingulate cortex, the greater the subjective effects. So we scratched our heads and think, what does this mean? We thought Leary was right. These drugs turned on the brain, but they don't, they turn off the brain. But how can turning off the brain translate into these experiences? And one way of thinking about it is to understand the organization of the brain in terms of the connectome, how different parts of the brain work together. And it turns out there are two major hubs, connector hubs in the brain, that have been identified in terms of connectomics. And they're shown here. The top one, the anterior cingulate, integrates your memories, your emotions, your planning, your anticipation, your executive function. And the bottom one, at the back here, the posterior cingulate, integrates all your sensations, your seeing, your hearing, your feeling, your proprioception. So the bottom one puts you together as an entity, and the top one integrates your understanding of yourself. And psychedelics dampen those connector hubs down. And so we thought, well, maybe what's going on, maybe the analogy is like an orchestra. The brain is like an orchestra. You have a conductor, you have two conductors in fact, but they talk to each other. You take the conductor away, then the individual parts of the brain do their own thing. It's like what happened when music changed when people stopped having conductors and stopped playing Mozart and they started playing jazz. You can syncopate, you can do things differently. And so we went back to this quote of Huxley. And this analysis that he made of the effects of psychedelics were that they lower the efficiency of the brain as an instrument of focusing the mind. And that's exactly what we've shown. And it was very pleasing to discover that neuroscience has eventually caught up with literature. And for those of you, just to quickly, so what is going on with psychedelics is this reconstruction of the visual images and also the sensory images and etc., all those different constructs that the brain makes, is disrupted by psychedelics. Because the control centers that build it up are disengaged. So what you're seeing, and this is truly, I think, remarkable, when you are seeing hallucinations, primary hallucinations, lights and colors and squiggles and that, you are actually seeing the primary workings of your brain. You're seeing what the brain sees before the rest of the brain turns it into an image. And I find that extraordinarily sweet. And we know this is true. We know because we know from complicated maths and physics of how those cortical columns in the visual system integrate information. We know that this is how the brain starts to work. And then eventually these complex shapes are gradually morphed into an image that you see. So I think we've kind of rather beautifully proven that not only Huxley was right, but also that visual physiologists were right. And in fact, just this week, a paper in Cell showing that in the mouse, this is what happens. If you give a mouse a 2A agonist, its visual system cannot construct a high-level image. It only gets stuck at this level. But there's another interesting aspect about this, which is those areas of the brain which show the biggest shut-off turn out to be what we now know to be the default mode network. Now, the default mode network is a group, integrated set of brain regions which work together to create the sense of self. No one knew anything about it. We didn't know it existed until we had brain imaging. And it emerged when we started imaging people who weren't doing things. Because the default mode is those parts of the brain which are active when you're not doing things. Now, all of you now are very interested in what I'm saying, and you're listening to me, and you're watching me, and so your default mode is switched off. But as soon as you drift off and start thinking about where you're going to go after this talk, your default mode comes on. So here's the default mode: anterior, posterior, cingulate, lateral, parietal. And under psilocybin, the default mode, which is normally strongly connected, the front and back, is completely disconnected. And that disruption of the default mode turns out to be very important for what I'm going to tell you later in relation to depression. Actually, when we did the first fMRI study and showed it switched off, we didn't believe it. We thought maybe this is an artifact. So we repeated the whole study using a different technique called BOLD, got the same results. So we tried to publish it. And we got some remarkable feedback from the referees. One of them said, it's all just blood flow changes from 5-HT receptors on blood vessels. Well, firstly, we corrected for blood flow changes, and secondly, the receptors are on, the receptors that are stimulated aren't on blood vessels particularly, and also, why would it be so localized? And then this one, what do you expect if you mess with the brain with psychedelics? Now, I'd expect better refereeing, frankly. But this is one of the problems, and this is the problem you guys will meet. The scientific establishment is not welcoming this for a number of reasons that we can go into in the discussion. But this perturbs the conventional linear way of thinking about the brain. And people don't want to have to reformulate their theories. And this is the quote. How many of you are scientists? So remember this and stick it over your desk. Orthodoxy is the diehard of the world of thought. It learns not, and neither can it forget. Now that wasn't written about science, it was written about politics, but it is so true for science. Orthodoxy is actually the death of science, because you end up just going into deeper and deeper self-referential kinds of circles. You've got to be unorthodox. Well, we had to answer this question, it's blood flow, so we did it by going and using a technique called MEG, magnetoencephalography. Now, magnetoencephalography measures the changes in magnetic circuits in the brain which are produced by changes in electrical activity in the brain, independent of blood flow. So there it is, and it's got fantastic temporal resolution. So we did the third experiment, we did psilocybin in people sitting up in a MEG, and we found exactly the same thing. The way MEG is analysed, you analyse it in terms of the power of a range of different frequencies, and across the 4 frequencies you can see that the posterior regions of the brain, the posterior cingulate, is powerfully disrupted. And at the high frequency, the 40 hertz, the beta 30 to 40 hertz, the anterior and posterior regions are disrupted. And there's a very strong relationship between the disruption in those parietal, posterior cingulate parietal regions and the ego disintegration, which makes perfect sense, because your sense of ego, or where you are in relationship to the world, is determined by your posterior cingulate cortex. And if you disrupt functioning there, then you aren't as you were before. One other really cute thing about MEG is it's got fantastic time resolution, milliseconds. So we were able then to test a theory that had been developed by neuroscientists, computing experts like Friston, that the oscillatory circuits in the cortex are driven by 2 excitatory cells and 2 inhibitory cells, interneurons and pyramidal cells. And you see the pyramidal cells here and the inhibitory cells here. And they've created theories as to what would happen if you disrupted those. And it turns out that the biggest impact we had was on these cells, which is exactly right, because that's where the receptors are. And I think that's actually the first time in human history that anyone has been able to say that in the human brain, a particular receptor, when stimulated, produces an effect on a particular. So we've actually identified a neuronal signature for a receptor interaction in the brain, which I don't think anyone's ever done before. But other interesting things came out of this. When you have a data set or data sets which are unique, you find there's a lot of people out there called biomathematicians, and they want to work with you. And we've worked with 3 different sets of them. And this I'm showing you because this is the prettiest one. And this is a technique called homologic scaffolds. And it's a statistical technique when it basically looks at correlations in the brain under the 2 states, on the left under placebo, on the right under psilocybin. Now they're constrained, there's 7,200 connections in each of those, but they look very different, don't they? They look very different because on the left, most of the connections are around the edge. That's what's called the small brain construct. Your brain works like that. Most of what you do, most of your brain's effort is talking to itself. Local areas talk to themselves, because that's the most efficient way of an energy minimizing way of living. Under psychedelics, there's much, much more crosstalk. And that increased crosstalk, I believe, is why people have novel, often very important experiences under psychedelics. They see things which they haven't ever seen before, because their brain has never let them think like that before, because the brain has got so locked into this kind of primitive small brain rituals. Well, having done 3 experiments with psilocybin perfectly safely with no problems, we thought someone's going to have to do the LSD experiment. It's never been done before. So there's Albert with his molecule, there's his original structure, and LSD has slowly morphed from a chemical to an art form, and tattoo and t-shirt and jewelry. I don't suppose any other drug apart from cannabis has ever become such a part of art. So we did the first ever brain imaging study with LSD, and actually LSD it's difficult to work with long acting, but that long acting and nature gave us an opportunity to do both MEG and fMRI in the same people on the same day. And we published this in PNAS a couple of years ago. And it essentially shows that LSD does what psilocybin does. It disrupts activity across the brain, and particularly here in the default mode network, the one I'm showing you here. So all those other networks are the other networks of the brain which we can look at, but the default mode network was fundamentally disrupted by LSD, and the more it was disrupted, the greater the sense of ego dissolution, exactly as with psilocybin. And we were able to do some slightly more interesting and elegant images looking at connectivity between parts of the brain. There's connectivity here between the parahippocampal gyrus, which is the gateway to memory, and the posterior cingulate. And again, the more you perturb those systems, the more experiences you get. And also we looked at the visual experiences of LSD and could show that there was a very powerful relationship between the increased eyes closed visual hallucinations and connectivity of the visual cortex. So let me just show it to you on this image. On the left hand side, this is your visual cortex now. Your visual cortex is being very, very efficient. Mostly it's just the bit that goes into your eyes is that blue area, that's called V1, that's where your primary input goes in. And your visual cortex is just talking to itself, really. It's not very interested. Most of you aren't even downloading these images to your hippocampus, but those of you who are will have a bit of connectivity here. But under LSD, you will see much, much more interesting and vivid imagery, because under LSD, the visual cortex is connected to everywhere in the brain. And that is why people can see things, but they can also see things like sounds and touch, etc. as well. The MEG results showed very, very similar findings to the psilocybin. Massive dysregulation of synchronized activity. Exactly the opposite of what we'd shown 30 years ago with the antagonist. So the antagonist synchronizes the brain, which is why you go into deep sleep. The agonist desynchronizes the brain, which is why you have the opportunity to think differently. And one of the most powerful effects is to change the power of the alpha frequency in the brain, which a lot of people think has got something to do with consciousness. It's an interesting area of research at present. I just want to show you, we've done a lot of MEG work. I like MEG because it gives you a kind of fingerprint of the brain. And you can fingerprint drugs with MEG. You can see how different these different drugs are. On the left-hand side, you've got a range of different inhibitory drugs. The sleeping pill, Zolpidem, and then one or two other drugs you won't have heard of which change GABA function. Then the anesthetic propofol, and then alcohol, which increases the power of across the whole frequency range. And then on the right-hand side, you've got LSD, and you've got psilocybin. You've also got ketamine. And ketamine produces a state of disorganized brain function that is not dissimilar to that of psychedelics. And that may be why also ketamine has utility in treating depression, as I'll show you the psychedelics have. And from that, we've come up, or at least I've kind of put out there a new two-dimensional vision of cortical function in consciousness. On the left-hand side, the red, that's the conventional model of consciousness, which is a glutamatergic model. You increase glutamate, you increase awakeness, alertness, you lay down memories, and it produces what I call parcellation. Your brain is awesome at defining very, very subtle differences in things. The fact, I don't know how many gigabytes of memory you're using for this talk, but it's tens of gigabytes. And you're remembering every single little fact. It is unbelievable how good your brain is at parcellating. Fact. On the right-hand side, the green, under psychedelics, you can still remember everything because you're not perturbing the glutamate system, but the way you are using the information is quite different. Psychedelics give valence to it, good or bad. They give meaning to your insights and your memories. And they also produce what we call integration. People can begin to integrate memories with feelings, facts with emotions, etc. So I think the psychedelic state is a separate form of consciousness. They obviously intersect because you can remember, most people do remember lots about their trips. We've also now started to look at ayahuasca, or the active ingredient DMT. And DMT is hard to work with. We can't use ayahuasca because it's a plant product. We don't know how to make it in a reliable way. So we use intravenous DMT. And what you see on the left-hand side is the dotted lines are the time course of DMT in the blood. And the colors, the yellow colors, are the power of the frequency of the EEG. And you can see the yellow band is actually the alpha band, the 10 hertz. And you can see that as soon as DMT gets in the brain, every frequency is disrupted. It completely, just the same way as psilocybin and LSD. So these three drugs we think all work in the same way. They powerfully disrupt synchronicity in the brain. They produce a state that people call entropy. Now what's interesting about DMT is that many of the people we put into this study have used DMT. Everyone's used it before. We wouldn't give people DMT in a scanner if they'd never used it before. They could have a very unpleasant reaction. But many of them like DMT because it takes them to a dimension which is different to the present, and often they find more real and more interesting. And often they see or they meet entities when they're there, and we're very interested in knowing whether those entities are in their brain or whether they're not. And so what we've done is we've asked people to draw pictures of what they saw when they went into the experience. And this is a dose response. We were trying to find the right dose of DMT to give. We now give 18 milligrams. And you see that the more DMT you give people, the more complex the images become. And at the very higher dose, you see that at least this particular person started to draw images of people rather than just of shapes. So this is a fascinating finding. Why the more you disintegrate the cortical integration, the more likely you are to see things which are actually like humans. And that's an interesting challenge to us, and we're going to have to explore. We haven't done the imaging yet. We're doing the imaging now. It may be that we can see, we'll be able to get more insights into to that when we've got the fMRI images. One other thing that I find fascinating, and we've started doing surveys on this now, is this relationship between psychedelics and colour vision. I got interested because some of the people who've been in our studies, they said, it's funny, but my colour vision's improved since you gave me that psilocybin. And people started writing to us, and so here is a very, very colourful and beautiful image. This is Monet's picture of Venice. And what you see next is an email from someone. All my life I suffered from red dichromacy, protanopia. So this is a relatively rare form of colour blindness. A painting, that painting, which I'd previously seen as a dull mass of brown and blue, all of the colours I was previously unable to see were there on the screen. And the emotion I felt made me unable to speak for about half an hour. So he was taken to this museum after he'd taken psilocybin by his brother. And for the first time he understood why his brother raved about this picture. It's not uncommon. As I say, we're writing up a paper looking at improvements in colour vision in people who've taken psychedelics. How can that be? Now when I talk to physiologists, it's impossible, because this form of colour blindness is in the eye. But it clearly isn't all in the eye. And I've actually dug out one paper which I think might be relevant, and this is that it seems that the brain isn't very interested in colours. And colour vision starts off being bilateral, and then it gets shunted away to one hemisphere. That's the small brain again. You know, colours actually, for most of us, are kind of irrelevant. You know, they don't define things in the same way as, say, a face does. And I'm wondering whether what you're doing with psychedelics is you're releasing the early ability, perhaps, of children before they get this suppression of colour vision to see things much brighter. So that's just an interesting thought for any of you who are interested. But the major clinical outcome from this research was our studies of mood. And we were fascinated by the fact that giving psilocybin to people in a brain scanner often resulted in them feeling better afterwards. And we were aware at the time that Roland Griffiths and his group at Johns Hopkins were taking people who were basically healthy, middle-aged people, a bit bored with life, wanting to make sense of life, and giving them a single psilocybin trip, the same doses we were using. And many of them were saying this was a very meaningful experience, and they were often having enduring, now 5-10 years improvements in well-being. So we thought, whoa, maybe psilocybin could be used to treat depression. And amazingly, we got a grant. The only money we've ever gotten from the government to study these drugs, they gave it to us because depression is such a huge problem. It's the largest cause of disability in the Western world. Getting the grant was easy. It was challenging then to do get the study approved. We had to go 3 times to the ethics committee, and in the end they said, it's too dangerous, you cannot give depressed people psilocybin. Why not? Well, it might kill them. It's pretty unlikely. Never been a death with psilocybin I've known of, and some of these people are probably depressed. In the end they said, no, you've got to do a safety study, you can't do a controlled trial, you've got to do a safety study, you've got to give 12 people psilocybin and follow them up for 6 months each, and if none of them suffer adverse effects, you can then do a controlled trial. And ethics committees have such power, there's nothing we can do. You either say yes or no, but I remember thinking, okay, but of course we must measure mood, mustn't we? Because maybe it gets worse. And they said, yes, you must measure mood, yes. So the mood outcomes are secondary, but the primary outcome was life or death. And then it took 30 months to get the drug. Only one place in the world would make the drug to a level which we can give to humans. And it took 30 months. Why? Because it's illegal, Schedule 1. Vast amounts of paperwork to make it and then to transport it into England. They were in Germany. And then another 2 months to get regulatory approval. So it took 32 months of a 36-month grant to do the study. I worked out it cost in the end about £1,500 per dose. And I speak to the regulators about this. Why do I have to have a special police check to work with psilocybin when as a doctor I can prescribe heroin? Why do I have to have a special safe to hold my psilocybin when the heroin is in the hospital safe? Why do I need all this? And they say, well, we want to stop you being a drug dealer. And I say, well, at £1,500 a dose, not even in Chelsea am I going to get any takers. It's absurd, but we have a bureaucracy, and the bureaucracy is about stopping research, not facilitating it. Anyway, we did the study, this feasibility study, 12 subjects, treatment-resistant major depression. They both failed on 2 previous drugs, and some had failed on over 10. They'd all failed on CBT. And we gave 2 doses, a low dose to make sure they didn't have a really bad reaction. They didn't, none of them did. And then we gave them the 25 milligram dose. And the effects were dramatic, way better than I could have hoped for. So every one got better, and some were cured. You get into the yellow line, in the yellow, you reach what's called remission. You're no longer depressed. So people who've been depressed for maybe 15 years are no longer depressed, and some have stayed well for months, and some have stayed well forever. That's the biggest impact of an intervention on depression, resistant depression, has ever been, after just a single dose of a psychedelic. But sadly, some people are slipping back. And the reasons for that are not clear. I believe it's because many of these people have been depressed for so long and have been traumatized prior to their depression so badly that their brain has learnt to be depressed. That is a kind of the default state of their mood is depression. And now we're looking to see if we should start giving maybe repeated doses to help people keep recovering. But we did imaging at the same time because we were fascinated by the question of how do these drugs, if they do change depression, do it? Now there's an enormous body of literature around the role of the amygdala in depression, and it relates to the sensitivity of the amygdala to fear or fearful faces. So here you see the amygdala, these 2 little areas here in the brain, these are the subconscious fear centers. If any of you see someone looking frightened, your amygdala will light up and will start to orchestrate you running away, because if someone's frightened, you should be frightened. And you don't want to ask questions about why they're frightened, because you might then be eaten by the tiger. So the amygdala activates subconscious fear responses. And in depression, the amygdala is hyperactive. A lot of studies have shown that. And antidepressants like the SSRIs dampen down that activity. And they dampen it down so effectively. Here's the meta-analysis. You can see here the power of the SSRIs to suppress amygdala activity. And that's how they work. They stop people being stressed and allow people to recover from their depression. So what happens with psychedelics? Well, we did the fearful faces, we also did happy faces and neutral faces. We did it a day after the treatment. And we found exactly the opposite of what we and others would have predicted. We found that actually, after psilocybin, amygdala responses were enhanced, particularly to fearful faces, but also to happy faces. And the more they were enhanced, the better the clinical outcome. So this turns the whole construct of amygdala and depression on its head. You can get over depression by suppressing the amygdala, or with psychedelics, enhancing it. And we've come up with the concept there are actually two separate ways of lifting depression. That the antidepressants dampen down amygdala activity, whereas the psychedelics increase it. But also, the antidepressants have a mixed effect on happy faces, whereas psychedelics increase that. And we're thinking there are two different ways you can lift depression. You can either resist negative emotions or maybe change brain activity so that you can see the world in a different way. And here's one of the psychological changes which has occurred in our studies. Before treatment, patients had what's called pessimism bias. They see the world as a nasty, stressful, horrible place. They're actually right, it is. But that doesn't help you get better. You know, most of us have what's called an optimism bias, that you see the world as better than it is. And after treatment, the patients become optimistic like us. And it really changes their outlook. Here's one comment from one of our patients. My outlook has changed significantly. I'm more aware now it's pointless to get wrapped up in endless negativity. I feel as if I've seen a much clearer picture. And many of our patients talk about defragging the hard drive, rewriting or overwriting the depressive ruminations, etc. And here's our new theory. This is a free download if you want to get it from the Journal of Psychopharmacology. We now believe there are two ways of lifting depression. The first way is to target the limbic system, that system deep in the brain, primitive system, known in all mammals. And that is by increasing serotonin to stimulate the 5-HT1A receptor, which is largely located in the limbic system. Dampening down, that's an inhibitory receptor, that dampens down limbic activity, dampens down amygdala activity, and protects you. Antidepressants give you a kind of force field. They take away the stresses of life so that your brain can heal. Whereas the 5-HT2A receptor stimulation works in the cortex, where you do your thinking. It disrupts rigid, inflexible, pessimistic rumination that you see in depression. And afterwards you reformulate your approach to life so that you have improved well-being, and that's why the depression disappears. And I'm going to finish with just two quotes. One from George Bernard Shaw, which is a brilliant quote for this work. Those who cannot change their minds cannot change anything. And I hope Michael Pollan's book was actually taken from this quote. I'm not sure it was, but if you haven't read Michael Pollan's book, How to Change Your Mind, it's a beautiful, beautifully written book, and it's a wonderful history and testimony to the value of psychedelics. But I'm going to finish with a political comment. Now, this is another quote from Huxley, and this is again not about science, it's about politics. But this is why it's so relevant. He said, greatest truth, but still greater from a practical point of view, is silence about truth. By simply not mentioning certain subjects, totalitarian propagandists have influenced opinion much more effectively than they could have by the most eloquent denunciations. And that is exactly the philosophy behind drug policy. Putting the psychedelic drugs into Schedule 1 was a deliberate ploy to stop people working on them, and if people don't work on them, you can't talk about them. So, well, it's been pretty effective. The block has been there for 50 years, but we're beginning to overturn it, and it's fantastic to see organisations like yours, growing in universities around the UK and the USA. So let's try to continue this debate. Let's have an open debate and have a rational and enlightened approach to psychedelic research. Thank you very much. Okay, what do you want to do now?
So we'll do Q&A. Alex,
Good.
Alex has a master plan. We can do some Q&A.
Take some microphones around. Good. Yeah, please do.
Let's do a Q&A. Can everyone can I put out a microphone?
But if you want to record it, you have to have the mic. It won't go onto there. If you want the questions on the film, you have. Ah, there you go. You got it. Yeah.
Okay, cool. Okay.
Yeah, so my question is whether you place the importance of testing conditions that relate to the valence of a psychedelic experience, how positive and negative is, if that's something that should be prioritized with current research, to try to start figuring out how to minimize bad experiences.
Yeah, hugely important question. So when we've looked at our predictive data from the first 20 depression subjects, good outcome was predicted by having a breakthrough kind of experience, a powerful experience. Bad outcome was predicted by anxiety. That was the one negative predictor, and it is a concern to me. I kind of wonder if the ancient Greeks got it right. Maybe alcohol and psychedelics, maybe, I don't know. I do wonder about whether we could take away some of the anxiety with a benzodiazepine. Obviously, we try to minimize anxiety, and we have a lot of preparation. Everyone has an education session, preparation session. There's always 2 therapists with them. So we do everything we can to minimize anxiety. But of course, the drugs themselves they have some tachycardic effects, so that you get a peripheral sensation, which some people find anxiogenic. And of course, a lot of people are scared about changing the way they think, and they're worried. We've had 50 years of lies. Some of them, people think, well, surely maybe one person could have a bad trip, and that could be me. So there's always a sort of subjective fear, and overcoming that is a big challenge for us. And I don't know the best way of doing it. And if you've got any inspirations, let me know. But that's something we really, would like to minimize, to maximize our outcomes.
And do you expect research on physical environments also, just whether high ceilings or low ceilings?
Well, so we always use music. We found that music probably has some bearing, but that in itself is complicated because some people don't like the music. And if they don't like the music, they get irritated. So that music generally is positive, but not always. So we're trying to see if we can tailor the music to the individual desires, which I think would be more sensible. Yeah, the environment, we always use the most comforting, protective, warm, embracing environments. But unfortunately, it's quite difficult for us because we have to do it in a hospital. So we have to, but they've given us a room. We've managed to been able to create a room in which we can do this therapy without having all the paraphernalia of steels, machines and things. So, yeah, I think it's extremely likely that the setting is going to be important. History tells us it is. And I'll just remind you of one of the studies, the American Psychiatric Association, not necessarily the most rigorous scientists in the world, when they wanted to destroy, one of the other pieces of evidence that the CIA used to get rid of LSD was a study that was given the study of the year by the American Psychiatric Association, I think in 1964. What did they do? They took people, chained them to their beds, and gave them LSD, and said it didn't work. And that was justification that LSD didn't have an effect. Outrageous, ethically immoral study supported, the American psychiatrists said it was the study of the year because it showed that LSD didn't work. So yes, so we know that really negative settings are very bad for outcome.
Great talk, Dr. Nutt. Two quick questions. In your LSD studies, what doses of LSD were you working with the subject?
So in the LSD study, we gave 70 micrograms intravenously. So probably equivalent to about 100 orally, probably.
Okay. And then in your psilocybin study, was there any analysis of the content of the experience?
Yes.
And the outcome?
Yes, there's a beautiful paper. I mean, I never thought, I should have put the reference up, I never thought that when I started this research I'd ever publish in the Journal of Humanistic Psychology. But it's probably one of my best ever papers. So there's a paper called Watts et al., Ros Watts et al., Journal of Humanistic Psychology, I think last year or early the year before, and it's got all the narratives, and they are remarkable and beautiful, and they show several experiences which seem to predict good outcome. One of them is connectedness. In fact, that seems to be the overriding, the positive connectedness with self, connectedness with the world, other people, seems to be a very powerful, powerfully associated with outcome. Yeah, but you should read it. It's a lovely paper.
Hey, so in reaction to your study now of visual reactivity to faces, obviously there's large comorbidity between depression and anxiety.
Yeah.
Were you able to partition out these effects of anxiety, and did you exclude participants who scored high in anxiety?
No, so these were all people with resistant depression. They all had anxiety. I mean, inevitably the two overlap. We didn't exclude, they all were primary depression, but, well, they've been depressed for years, so I guess, that's what they had I don't know whether some of them started off being anxious 30 years ago. It's a very important question. We get asked this a lot. Would I countenance? Is anxiety a topic, an area that we should be trying to treat? And I'm not sure. So there are forms of anxiety like panic disorder where I can't see why psychedelics might work. But there are forms of anxiety like OCD where I think I could see why they could work. So at present we're exploring the possibility of doing an OCD study. The problem is that the patient groups we talk to about this, they have said categorically they are never going to have an experience which they're not in control of. The idea of actually losing control and going somewhere else in the universe for a bit is not something they would ever countenance. So we're compromising at present. So we're going to try a 10 milligram dose to show that they won't come to harm, and also possibly that might be like psycholytic psychotherapy. It might help them engage in the psychotherapy, the behavioral therapy that often they're quite resistant to do anyway. So we're having a sort of compromise there because our patient group will not tolerate. Well, they want at present they're refusing to consider having a high dose.
Hi. So actually I have one question about the hallucinations.
Yeah.
You were talking about how your brain essentially can't put together the simple visual stimuli, and it can't like stitch together to a complex image of like what the world is. But you also showed that the thalamus gets
inhibited.
On the sector when you're on psychedelics.
Yeah.
And since the thalamus is kind of responsible for gating your sensory input, I'm wondering how much of a hallucination do you think is an inability of your brain being able to create that a more like real image, and how much of it is you just have more sensory stimuli than you're used to, and it's just all kind of like overlaying on top of it itself.
Yeah, it's an interesting question. We were surprised at the thalamus. We were surprised at everything, but we were particularly surprised at the thalamus because it doesn't have any receptors, and so I presume that's a secondary effect. It's presumably either there's some top-down inhibition going on, or we're taking off top-down excitation to lead to the thalamus being attenuated. It's difficult to know. That was more profound in the psilocybin studies where we gave it intravenously than in the studies where we give the drugs orally. So it might be something to do with the speed of change, which can be. So I guess we'd expect to see it. We can't see that when we look at the DMT study. It'll be interesting to see the thalamus, which is off there. Yeah, it's the way I conceptualize. I mean, the thalamus is clearly an important sensory station for getting input into the brain. So switching it off could cause alterations in consciousness as well. I mean, the literature suggests that if you turn off the thalamus, you actually go into coma rather than go into a state of increased awareness. So but the reason I'm going on about this is there's actually a debate in the literature at present, because the other group that's done a lot of the kind of comparable work with us on psychedelics is Franz Vollenweider's group in Zurich, and he's got a thalamic theory, which I think is wrong. He thinks that the thalamus is central to these psychedelic experiences, whereas I think these are probably, the changes in the thalamus are probably not artifactual, but secondary to the primary changes in the cortex. Are you right?
Yeah, so is there music playing? Or
Yeah, there's music playing from up here.
is that bothering you? Okay.
No, I'm glad you can hear it too. I was getting a bit worried.
For a second I thought I was hallucinating.
Hi, thanks for your talk. It was really interesting. My question is about, has there been any research out there on how hallucinogens can affect long-term neuroplasticity and really rewire the brain to get to sort of a more open state?
Yes. Certainly there has been. I mean, there was a couple of animal studies now showing you can get plasticity, you can get synaptic spines growing. I mean, it's quite compelling. The animal literature is quite compelling, and that's kind of we've assumed that is what is happening. We assumed from the very start that these receptors wouldn't exist if they didn't have some role. But the really interesting question is why they're not doing much in the normal person. I can block all these receptors in your brain now, and you wouldn't notice. So why are they there? And we're thinking that they're there either because they're really important in growth, in development, in helping the child organize its thinking, or that when you confront something that is really important to you, where you have to make a life change, then they facilitate that, or they enable that. And without them, you can't make those fundamental changes in your life, which are necessary for you to, cook your food rather than just eat the meat raw. That kind of thing. It may be that they are critical to the sort of step changes in cognitive and attitudinal functions that people need to engage with or to embrace if humans are going to evolve in the most appropriate way.
Hi. First, I just want to thank you for a really amazing talk and really brilliant research. So to kind of derail a little bit, just because just some things I've been thinking over the past day and a half now, I'm just curious if you know of any research of the effects of psychedelics on immunology, in particular neuroimmunology, because I was thinking of it as an interesting model of a physiological model rather of the internal versus external barrier, because the immune system is there to define self and not self, and it fights infections and it protects your internal body. So kind of thinking of is whether or not practitioners or people that are that use psychedelics, there's differences in their immune system function and how well they respond.
Well, they live to 100, don't they? So it must. Clearly, they're not just. But seriously, it's a great question, and one could do the experiment. One could take people and one could give them psychedelics, and then you could test for their ability to react to flu vaccine and see if they get better antibody production, just in the same way as, those experiments have been well done now, showing that stress reduces the ability to mount an immune response to things like flu vaccine. So one could do that experiment. But there's a whole possibility that there are peripheral effects. Actually, there's a whole branch of, you can't really call it psychedelic research because it's not psychedelic, but there's a branch of using low-dose psychedelics to try to modulate immune function. And there's a study going on to see whether you can treat asthma with of these drugs. So that is a field which is likely to grow in the next few years.
Sorry, just quickly, do you know who were doing this study? I'd love to read more about that.
Say again?
Who is doing this study?
Yeah, well, I do, but I think it's confidential, so I don't know if I'm supposed to know. So, but just, I mean, I think if you look, I don't even know if it's on clinicaltrials.gov. I suspect it isn't because I think it's commercially confidential. But you know, it just gives you some hope. You know, I just wanted you to know that people are thinking. You know, you are not alone.
Okay, so I know there's been relatively little research on psychedelics.
Could someone wave? I'd like to see you. Ah, good, thanks.
Yes, I know there's been relatively little research on psychedelics mood disorders. I'm wondering if you either done any work or if there's anything in the pipeline that you know of looking at predictors, either clinical, psychologic, or imaging predictors of treatment response.
Yeah, so I showed you the predictor of amygdala reactivity, predicted treatment response. We've now subsequently shown that change is due to a change in connectivity within the prefrontal cortex and the amygdala. And there was a paper in Scientific Reports last year or so on that. Those are the two predictors we have at present. And if you have a more powerful experience, is more likely to give you a better outcome. So that paper's also been published recently. That's Roseman as the first author. It's quite difficult though to be sure about those things because you never know whether that's just a dose effect, because obviously they all get the same dose but different amounts get in their brain, so we can't be absolutely sure. It's not a sort of an artifact of just having more dose, but it's kind of plausible that the more psychological experience you get, the better the outcome. It would be surprising if it didn't.
You didn't find degree of treatment resistance was predictive at all?
No. Well, they're all pretty treatment resistant. And you can see, I mean, if you just go back, you can see that, you know, look where they all are there. You know, there's not a lot of range, you know, I mean, they're all over 20, you know, no, there wasn't, and the numbers are too small to, there wasn't a relationship there, no.
Thank you.
I have two more, but we can go off on a little bit.
I got to leave at noon, so that's fine. Just carry on.
So I noticed that a lot of the studies kind of fall under the categories of classical psychedelics like LSD, DMT, mescaline, and psilocybin.
Yeah.
So I was wondering, are there currently like studies in the works to more intimately understand the vast diversity of psychedelics that exist, just the hundreds of chemicals out there, some with very unique effects like exclusively auditory effects? I know there was a fairly obscure one that was described as an antidepressant in the Soviet Union, alpha-ethyltryptamine.
Yeah.
And I know there's like kind of a limited amount of resources going into psychedelic studies. Do you think that's kind of like
I think the answer is none. Limited is none. No governments are funding this. I mean, I think it'd be fantastic to do a proper detailed analysis of different kinds of psych. I quite agree with you, but that's not going to happen. So what we have to do is and that's one of the things we are doing, is trying to collate evidence from people who are using different kinds of psychedelic to see whether we can generate enough anecdotal data to justify writing grants. So that's why you've all got to log in, and when the Global Drug Survey comes out, get on there and answer the questions, because we can use that. So the data I'm going to write up soon on colour vision, that's all come out of the Global Drug Survey, just asking that question. Now we've got a huge data set of people whose colour vision has improved. But I don't think the Global Drug Survey has even thought about looking at these other ones, and that will be a good thing. So if you email me afterwards, I'll pass it forward to Adam Winstock, and we can maybe put that in. That's quite an interesting question. You know, whether some of these rare psychedelics with their peculiar or reported peculiar impact, because it would be nice to know. You know, because the danger is if only a few people have used them, it might be the person rather than the drug that makes it odd.
Thank you for your talk. I had a question in regards to the music that you choose for the psychedelic.
Yeah.
based on work that you did with Dr. Mendel Kaelin? You can talk a little bit more about that.
Yeah, so Mendel put the work, yeah, and that was his PhD. He put the music list together. So he created the music list. But some of it's quite dark. I mean, I don't know if you know the Symphony of Sorrowful Songs by Goretzky. I didn't think that was a wise thing to put in. I don't think he quite understood what it was about, which is about the Holocaust. And I don't think he understood that. So we're taking that out now, because as much as it's a wonderful piece of music, it is actually about people dying. So I think some of our patients knew that and he didn't. So yeah.
So my question is addressing you in your former role as a drug policy advisor to the UK government. And I'm interested in how you would expect drug policy and regulation to change perhaps over the next 5 years, and whether you see differences with regards to the US and Europe, UK.
Yeah, great question. So my hope is it'll change fast. My expectation is it'll change slowly. I think the US has, the great thing about the US is that the states, you have two bites at the apple. The feds aren't going to change, but the states can change. Now I believe Denver is going to try to make mushrooms available. I think Oregon is going to try to put mushrooms in their next election as to decriminalize them at least. So I'm really hopeful that 1 or 2 American states might change, and that would give a lot of impetus to the rest of the world to change. I mean, the one good piece of news is that some kind of mushrooms are available in the Netherlands. The Dutch have been really very wise in this field, and when in 1971 the UN banned psilocybin and the psilocybin mushroom, the Dutch banned the mushroom, but they didn't ban the truffle underground, which it grows from. So it's always been legal in the Netherlands, and actually that's actually become a huge business now. So if we have patients that we can't treat, we say, well, if you want to get treatment, you go to the Netherlands, and many of our therapists go there to get experience themselves. So but my aspirations are to get psilocybin descheduled, hopefully in my lifetime, but I'm 68 nearly, so we got to get a move on, right? So you work harder, guys.
Thank you for the talk. My question was regarding, with Roth, it was mentioned in an earlier talk how his therapists were required to experience LSD themselves 5 times before they were ever going to actually monitor somebody else. Additionally, you mentioned that I was contracted for new arrivals to experience it themselves. And within like the different religious communities that utilize sacrament, such as in the case of the Native American Church, they're required to have very extensive experience before they can undertake the role as shaman.
Indeed.
Do you see that being beneficial and integrated in any way, shape, or form?
Yeah, I mean, it's a very, very important question. Well, the first thing I will say, it can never be directed. I mean, and that's one of the problems we had when it was being given to trainee psychiatrists, because some of them didn't want it and had a bad trip and started suing the government. And we don't want that, so it has to be volitional. And then the problem is with illegal drugs. It's quite dangerous for doctors to start talking about having taken illegal drugs. I mean, because you could be struck off the medical. That in itself you could be struck off just for saying that. So we have to tread very carefully, and I think, but certainly it's quite likely that personal experience will have some benefits, but not necessarily. Because one of the points about, people have very individual reactions, and it's conceivable that your reaction might be different to other people's. And most of the therapists aren't depressed. So it's not intuitively necessarily the case that the experience of a non-depressed person would be that useful to a depressed person. So I'm open-minded. I would certainly not discourage it, but we certainly could never commandeer it or command it.
And I had just kind of a segue off of that.
Yeah.
Regarding the ritual around that.
Yeah.
Part in that. How have you integrated a ritual in your studies and been able to create that second stack?
Well, we do have we don't call it a ritual, but we call it good clinical practice. But you're right, it is a kind of ritual, and it's about selection and excluding people who're going to have problems. It's about preparation, which is half a day. It's about the session, and we don't. With the psychedelic studies, and we're doing an MDMA study as well, where we do actually engage with the person through the MDMA for PTSD. But with the psychedelics, we make no effort to engage. We're there if they want to engage, but they rarely do. And the real work is done the next day in the integration session when the two therapists are there with the patient, and then work through for a few hours the experiences. And then that working through obviously can continue for as long as they want or as long as they live. All right. So it is a kind of ritual. But in a very clinical setting, yeah. And it's a really interesting question. There are studies now of the effects of ayahuasca in terms of depression, and that's done in a much more ritualized way with shamans. I mean, maybe that's more powerful. I don't know. It'd be quite interesting to compare the two.
Thank you.
But there are people that don't want to go and be treated by shamans. Some people prefer to have the old psychiatrists like me.
Just one more question. So just to play devil's advocate a little bit, as a neuroscientist, so I'm speaking to I'm not a neuroscientist, but from what I understand, the main ways neuroscientists study the brains through imaging of the electrical connectivity brain, and through blood flow and oxygenation, and those are kind of the main ways of looking at again, just to play devil's advocate a little bit, do you think that, just ask you to speculate, do you think that there could be other modes of connectivity within the brain that the traditional scientific canon is not even asking the right questions to look at?
Well, there could be, but it'd be hard to study scientifically.
Sure, yeah. I'm just asking you in your experience.
Well, that's why we're looking for the entities, of course. And if they are out there, then that's great news, because that is a whole new, that's like relativity re-instruct. I don't know. I suspect they're probably not, but anyway, we'll find out.
Whose hands?
Good morning, Professor Nutt. I'm a big fan of your work and admire your will and the way you stand up to the establishment that it just goes off and interprets your studies.
Thank you.
My question is, with the epidemic of opioid addiction and heroin addiction in the United States, what do you think would be the steps for the public to be aware that there is an option in the form of medical game therapy? Do you think there would be at all a possibility to somehow make that treatment more on the radar of an average Joe opposed to...
Right, this is, So let me explain. So the question is about ibogaine for opiate dependence. So ibogaine is an interesting, it's another plant psychedelic. No one's ever imaged ibogaine, and the problem is no one's going to, unless if you've got some money, we'll image it for you. But no, seriously, I mean, it would be quite an interesting question to do the brain imaging of ibogaine. It's a difficult drug, really difficult drug to work with, because the trips last forever, like days, so that we'd have to have people in hospital. And so that's one of the reasons we've partly avoided it. The other reason we've avoided it is, well, we have no money. And the third is that it does cause cardiac changes. And those are dose related. So a colleague of mine, one of my trainees, Paul Glood's done a very nice ibogaine study in New Zealand showing that you've got to monitor the heart. So And Ibogaine, of course, has been associated with some death. So giving Ibogaine to heroin addicts withdrawing in the jungle is not a good thing. But in a hospital setting, it's potentially quite a useful thing to do. It is a legal medicine in New Zealand for opiate addiction, and I've spoken to people who've used it and treated themselves and been treated. And I'm open-minded. I think we should definitely not dismiss it, but we should try to study it. But as at present, it's not being studied. But that gets us to another question, which is really perhaps the real thing, the useful thing to do is not treat people who do have opiate addiction, but stop people getting opiate addiction in the first place. And we're in the process of trying to set up a psilocybin study for chronic pain, because we know that opiates don't really work very well in chronic pain, but once you've started using them, you can't stop. So we are in the process of developing a protocol to see if we can use psilocybin to help people with chronic pain not have chronic pain and potentially get off their opiates. Yeah?
Hi, thank you for being here. So, in regards to the depression research, you mentioned that people will sometimes come across insights that may inspire them to make fundamental changes.
Yes.
Could potentially help alleviate their depressive symptoms. And I was wondering what you think about the role of the environment in that, as far as constraining the ability to make those fundamental changes, as well as potentially creating the conditions for onset of depression in the first place. And maybe you could speak a little bit about the integration work we do as far as helping integrate the experience.
Yeah, okay. So that's a big question. There's a lot in it. Yeah, it's unfortunate that we have to treat depression that has been provoked by environmental disasters, social, usually interpersonal disasters, family, parenting disasters. It's a real problem. But as I say, I can only do so much. Give me a break, I can't change society. You've got to do that. So, yeah, we are always patching up the results of damage. But what we are doing here is that there are people with chronic depression and other chronic disorders where the damage has set them into a way of thinking which perpetuates the damage. And that's what we're trying to disrupt and change. But I kind of agree with you. Even if we were to take a heroin addict and we were to stop them craving heroin, it was amazing coming here yesterday, because I got the train from DC, and it went through Baltimore. And as I looked out, as we left Baltimore Station, there was the wire. Actually, it's worse than in the wire now. Downtown Baltimore is even worse than in the wire. There's almost no houses that people are living in. And I thought, there's no point. You can't, stopping someone craving heroin and putting him back in that environment is probably pointless. You've got to have ways in which people can engage with life in a way that can change, was different to what they did before. So whatever we do, it's got to be integrated into a proper holistic form that can help people be empowered to do the things they really need to do, that can protect them from subsequent relapses. Yeah.
We have time for 2 more questions.
But after that, we have it's like people still figuring out. So 2 more questions. So back there.
I have a question, by the way. When you give it, when he's asking mine, I'd like another coffee, please. And then when you finish the 2 questions, I'm going to sit here, and the rest of you can, if you want other questions, you can come and ask me. I'm going to sit here, drink my coffee, and have my donut. All right? Carry on.
Hi. What do you think about the...
Wave, I can't see you.
Oh, I'm here.
Yeah.
What do you think about the idea that the visual geometries you see on DMT reflect the inner architecture of the mind?
Well, I think everything we're seeing is reflecting the inner architecture of the mind. Yeah. But the mind is not a one thing. Your brain is the most sophisticated. Maybe you don't. Your brain has more computing power than all the computers on Earth together. And you can't talk about, there being one computer. That what we're looking at is the mind is an enormous complexity of different processes, and you certainly see quite a number of them when you give people psychedelics. But there's not a mind. I mean, it's a kind of strange, unless you think. I mean, I don't understand. How could there be a single mind? What does that mean? What are you talking about? I mean, I don't see how you could do science on the concept of a single. What did you mean?
Also, one idea is that one idea is that, for instance, there have been these neuroscientists lately who've been trying to extend the concept of grid cells from just physical space to abstract conceptual space.
Of which cells?
Grid cells.
Yes. Yes.
So I'm wondering whether, like the sort of visual geometries that you see on DMT reflect the way in which the brain perhaps breaks down abstract conceptual space or other kinds of space.
That's a great question. A great question, yeah. And it's very likely. And these grid cell people should be giving their animals psychedelics, shouldn't they? It would be, they should be. They absolutely should be. And that's why the paper in Cell this week is really nice, because they've done that for primary visual cortex. And it's really elegant. So read it. Any more?
Last question.
These have been really awesome questions.
So a lot of your studies have looked at depressive symptoms, and classically use DSM to diagnose these things, but there's this big push now to use the research domain criteria, cognitive frameworks, and
absolutely,
rather than symptom-based diagnoses. I was wondering if you have any plans to kind of integrate this framework in your future studies and what you make of it in the context of psychotherapy.
No, it's a really, I'm a great fan of the dimensional construct. I've always treated my patients as having dimensional problems rather than diagnosis. Diagnosis exists for billing. They don't exist in the real brain. And it would be interesting to look at the ones who are slipping back here to see whether there is a dimensional difference between those and those that don't. It would absolutely. And we have the data to do that. We just haven't done it yet. Then the end's a bit small. So now just to finish, so you know what we are doing. Because I haven't mentioned this, we do have money from a charity now to do a replication of this study, and also a controlled comparison with escitalopram, the SSRI. So and in fact we're doing 2 doses. So we're doing psilocybin at time 0 and at week 3. The primary outcomes at week 6 when we compare that with 20 milligrams of escitalopram. And our primary outcome is actually, we predict the brains will be quite different in terms of the amygdala responsivity and the connectivity. That's our primary outcome. But we'll be able to look at other factors as well. So that's ongoing. We've recruited about 1/3 of the patients into that, and that'll roll out in 2020. All right? Okay.
Thank you so much for all the great questions.